Dear everyone, greetings from Asian Productivity Organization. This is live from the APO Secretariat office in Tokyo, Japan, and we welcome all our viewers from APO member countries and beyond. I am Asai Thambi Monikam, and thanks to our viewers for joining us on this APO Productivity Talk series. And today we will discuss about Agri Innovations from Bioprime. As climate change continues to impact global temperature, weather, and seasonality, long-standing agriculture practices are undermined, throwing many farmers into poverty. Subsequently, the world's rapidly growing population is ever more reliant on increasingly vulnerable food production. The sector must urgently adopt new technologies, products, and approaches are required to mitigate and adapt to climate change while supporting growth and jobs. Innovation and research development is really key in driving how you can grow foods. In today's scenario, many agri startups are trying to address the climate change issue in agriculture by offering innovative solutions or products by focusing on new innovative technologies which are also sustainable one. In, in this way, Bioprime Agri Solutions is one of the technology-driven startups from India which offers sustainable solutions in the form of agri-biologicals by leveraging biotechnology platforms. To explain further about Bioprime's innovative solutions, how it contributes for increasing productivity in agriculture. Today, I am very much delighted to welcome our guest speaker, Dr. Renuka Karandikar, the CEO of Bioprime Agri Solutions from India. How are you today, Dr. Renuka?
I'm doing very well. First of all, thank you so much for everybody at APO for inviting me here to share about the work that we do in Bioprime. I look forward to this session. Thank you so much.
Thank you so much for joining us today. Before we get into further detailed discussion on this topic, let me share a video clip from Bioprime. Have a look.
Bioprime is an ag-biotech startup based in Pune. Today, more than 70% of the agriculture is impacted by climate change. Unseasonal rains, increasing disease and pest attacks have led to ever-increasing costs in inputs. However, the productivity and farmer income decreases continuously. The most severely affected amongst this are the small and marginal holding farmers.
We are using natural molecules to trigger inherent capacity of crops to fight against changing climate and attains its highest growth and yield performance.
Plant algae microbes have an arsenal of biologically active molecules. These are isolated using optimized processes to retain them in the most active forms. Hundreds of such molecules constitute Bioprime's unique library of bioactive molecules.
Bioprime has developed proprietary rapid screening assays using cells and tissues. These assays help in evaluating hundreds of molecules in a matter of days and at a fraction of the cost. Carefully selected molecules modulate various physiological processes in crops to deliver targeted results. This is Bioprime's technology platform, SNIPR. High-throughput equipments and methods are used to ascertain quality of raw materials and finished products, maintaining batch-to-batch consistency.
SNIPR biologicals are crop-agnostic, certified organic and residue-free. They are formulated in different forms to ensure compatibility with existing agronomic practices and can be adopted by farmers on as-is, where-is basis.
These formulations induce resistance to biotic and abiotic stresses in crops. Various productivity parameters such as yield, size, color, sugar content, shelf life are successfully targeted. We are thus contributing towards doubling farmers' income and Atmanirbhar Bharat.
Before I invite Dr. Renuka to start her presentation, we would like to encourage our viewers to send your questions and comments in the chat box with your name and country, and we would respond to your questions during Q&A session. Dr. Renuka, now the floor is yours.
Thank you so much, Asai. Yeah. Uh, so today I will be speaking primarily about productivity in relation to climate change impact or the stress that the crops are facing. When we look at the scenario of climate change impacting the world today, we will see very clearly that some countries are being targeted more than the other countries, especially the Asian continent as well as the African continent and South America faces severe climate change impact than the other countries. So we are left to feed for ourselves and fend for this impact. If we look at how climate change is affecting productivity in different crops, we will see that, especially in large acreage crops such as corn or wheat or rice, we can expect 10 to 20% drop in productivity. Given the scenario that we're facing today, a single event like a war in Ukraine can lead to a catastrophic drop in the food security. For a very long time, though India was a primarily agriculture country, grains were not exported from India. Due to these conditions, suddenly countries like India have come into forefront and are looked upon to feed the world and become the breadbasket of the world. But how does India or how does the world navigate the problem of feeding ever increasing population on one hand and deal with robbing of yields and lessening of productivity on the other hand? If we look at the impact of what all of this means for people like us or consumers, we can see that in the past 10 years, we have seen a sudden rise in the prices of the food that we consume. If these scenarios are not met, which means the food is only going to get costlier and it is going to become more inaccessible, especially for the people at the bottom of the pyramid. It's not just about the food getting costlier, it's also about the nutrition this food provides. If you look at the nutrition that was there in food that were produced 10, 20, or even 30 years before, we will see that the amount of proteins, amount of sugars, amount of carbohydrates or vitamins that was derived from this food is much less compared to what we get today. I'm sure many of saying that the food tasted different, the food was more fulfilling, and it was more nutritious. On one hand, we face these challenges, and on the other hand, we face problems such as farmers not being able to get the same amount of productivity. Now, let's just look into why farmers are not being able to get the same amount of productivity. Today, agriculture faces a multiple of different challenges, whether it is heat stress, whether it is cold stress, whether it is short duration water shortages, or whether it is intensive radiations in the form of UVs. Mind you, this is not a challenge that happens, not like one of a challenge that happens in the entire life cycle of the plant. A crop goes through multiple of these challenges in a single crop cycle, and the farmer has to deal with the changing scenarios as he goes along farming. When we are looking at this, the current agri inputs become insufficient to mitigate these changes. Now let's look just very closely at why this is so. Have today basically make available nutrients if they are not present in the soil. When these are taken up, the plant uses its own machinery to convert this into food. Then this food is either stored or used up by the plant to grow, to flower, and to fruit. Now, what happens when the plants are facing stress conditions? What happens when the plants are facing a multitude of these stress conditions at a single time? It happens that the nutrients, it is not able to use these nutrients effectively and to produce the food. And this food is not being used in a more efficient way for growth, for flowering, and for fruiting. And what happens is that the farmer sees that the crops are not growing as per his expectations. His instinctive action is to add more nutrients, add more fertilizers, and he does not see the expected result. And he goes back and adds more nutrients and fertilizers. Now, you will realize that when I was describing this scenario, the problem is not unavailability of these resources. The problem is the plant's inability to use these resources. And this is something that the to address. Therefore comes a need for new kind of agri inputs, a new class of agri inputs. This is where Bioprime works in. Bioprime, as its name suggests, works on bio-derived molecules, microbes, or anything that is of natural origin. And prime has two meanings. Either it primes the crops towards perfect condition, a better condition, or it is to modulate different processes. In a nutshell, Bioprime covers the hypothesis of what we do in terms of our research and how we look at developing new kinds of agri input products. So, just to summarize what we discussed right now, the climate change or related stress impacts leads to overuse of fertilizers, which is spoiling the fields, which is spoiling the soil, and leads to more disease and pest attack, and therefore farmer has to use more insecticides and pesticides. This results in increasing cost of production, which ultimately consumers like us have to bear, and farmer is left with what we imagine at Bioprime is every harvest becomes a successful harvest. We imagine that for every crop that is sown, the farmer is able to reap the yields and the profitability that he desires and definitely is worthy of. What we do at Bioprime is we develop biologicals which are based on small biomolecules that target different processes in plants, or we look at different kind of microbes that by associating different traits. Now, for a minute, just let's look at the physiology of the stress. If you look at the extreme left-hand side figure, it will tell you the impact different kinds of stress has on plants. And what we want to cover here is just take you through the challenges that the farmer is facing on his field today. So he faces low moisture and problems with nutrient uptake, which leads to decreased photosynthesis, which leads to overmaturity or shorter life cycles of plants, which leads to affected reproductive stage, which means low flowering, low fruiting, or loss of flowering and fruiting, which may be in terms of quality and which may be in terms of quantity. And if we look at the level or the physiology that happens, it is covered on the extreme right hand side. So this gives us the target on which we want to work on. So for example, we would like to work on retaining moisture within the plant, ensuring that the photosynthesis efficiency is maintained in stress condition, ensuring that there's a sugar that is being translocated at the correct time to either the grain filling stage or the fruits and the flowering stage, so that the farmer is able to get a. So what does Bioprime products do? So if we just extrapolate from the stress physiology here, at the very low end, we see that the low moisture and the nutrient uptake is counteracted by two of our products, Prime Verdant and Prime Fortiseed. What Verdant and Fortiseed do is they protect and insulate the plants against changing climatic conditions. Verdant acts as a sugar and salt reservoir, making sure that the cells remain hydrated even in extreme heat. It also makes sure that the photosynthesis apparatus, that is the engine that is doing the photosynthesis. What Fortiseed does is it enables the plant to grow in low light low temperature or high light and high temperature kind of conditions. When we go to the next stage, that is altered sugar translocation, altered flowering and fruiting, or altered quality of the produce, that is where our product prime chiron helps in. What chirons does is it directly targets the flowering and fruiting pathway using signaling molecules, and it also modulates the sugar budgeting, which is the allocation of sugar resources to flowering and fruiting and grain filling stage. Uh, in combination of photacy and avidity right in the center, what the life of the plant, delay the senescence of the plant, and manage the stress condition in plants. On the right hand side, you can see the impact this product has in field. If you see the roots, the root ball, the root biomass is very good. The water holding capacity, the soil holding capacity of the treated plants on the right hand side is much better as compared to what you see on the left hand side. The biomass development is much better. Early plant establishment is obtained. When we look at the reproductive characters, you see that the ear heads are more head, and therefore, when we harvest, we get better productivity, more weight of the grains. So this is just an example to show how the products overcome different kind of climatic stress conditions. So these biologicals or these biomolecules are able to make crops climate resilient. Ultimately, the food that you eat is more nutritious at a cost of less nutrients that are put into the soils. Because the plants are healthy, insecticides and pesticides are required in small quantities. Using the same technologies, we're also pesticides. These products, soil restoration, and the food that we eat is at a lower carbon footprint than it was obtained previously. Thus, we have food security, we have sustainability, we have lower carbon footprint, and ultimately, we have residue-free food. This is the three philosophies on which Bioprime works, that is targeting climate change, reducing the fertilizer cost, ultimately meeting the consumer demand of having more nutritious, more residue-free food, and helping farmers adopt more regenerative practices. If we look at the agriculture biologicals that we have today in the market, we see a large portion of it is biofertilizers and biopesticides. The upcoming segment is biostimulants, and the newer segments that are coming up into the market thanks to the new scientific advancements are seed treatments, herbicides, and insecticides and pesticides. If we look at where most are conducted, we did a literature survey and mapped it against the country of origin and found out that Italy, Spain lead the biological research currently globally. Um, there's a lot of research that is coming out of India, Brazil, and US and China as well. If we were to look at what are the kind of areas that people are working in, then we see that a large number of non-microbial products are coming into the market. And one of the largest segment here is the protein hydrolysis segment. And we also see a lot of research that is being done on microalgae in comparison with the macroalgae that in the microbe-based segment, we see a large still occupied by the mycorrhiza and the trichoderma segment. But if we were to focus on what is it that we can build for the future, then we try to map different areas in which a large amount of attention is required and science driven products are required. So, if we see stress and nutrients still dominate the products that we see in the market today, as well as the research that is going on in the market, in the research laboratories, directly targeting and yield enhancement is still a very short percentage of this research. So a lot of focus can be given on yield enhancement, a lot of focus can be given on food fortification, and a new upcoming area for this research can be reducing carbon footprint. So from expertise, we identify these three areas as key target areas for future enhancement of or new kind of biological products that we will see in the next 5 to 10 years. Now, we would like to take you through some discoveries that are reshaping the biological sector. In primarily, I will be speaking about two platforms that we have developed here in Bioprime itself. We work on SNIPR, which is a biomolecule discovery platform, and we work on discovery platform. And I'd like to speak a little bit about this in a couple of slides. So SNIPR basically is an acronym, and it stands for Smart Nanomolecules Induced Physiological Response. And it's nothing but a multidisciplinary high throughput screening platform where we combine different approaches together. We have phenomics, we have physiology, we have biochemistry. In omics, we work on metabolomics and transcriptomics, and finally combine it with bioinformatics to arrive at certain leads for development of next generation of ag biologicals. Under BioNexus, trait modifying microbe library. Now, what I mean by trait modifying is there are certain microorganisms which, by association with plants, can confer certain traits to plants. So, heat tolerance, tolerance to drought, insect tolerance can be or are currently addressed as breeding techniques. So, researchers are working in terms of identifying parents and breeding newer varieties. Our approach is that by combining them with microbes, the current existing varieties can be upgraded to have these additional traits. Breeding requires 10 to 15 years before a variety hits the market. So, till that time, there has to be some ways for the farmers to mitigate the changes that they are facing in the fields today. And we see that microbes, these special kind of trait modifying microbes, is a way of giving their climate losses. So I'll just walk you through what we do in SNIPR and how a lead is identified. The two approaches that run parallelly. One is a high throughput screening approach and one is a high resolution approach. In high throughput screening approach, we are looking at a hit or miss kind of a lead identification, whether a molecule or whether a microbe is able to or not. That does not give information about what is the mode of action, how it is working. Once we have a high throughput approach and we have certain leads identified, these leads are then put into a high resolution approach where we understand more about the functioning, the mechanism of action, and get more deeper insights into these leads. For high throughput screening approach, we use biochemical and physiological methodologies. We use spectral analysis, we use cell and tissue analysis in the primary screening process. No plants are used in the screening process, which cuts down the time taken for screening from weeks to days or sometimes even to hours. Um, these are usually done in small microtiter plates and can be done as a 96 microtiter plate, and within a couple of hours this entire process can be completed, giving a in the high resolution approach, once we have quantified leads, then we work on identification of what is the pathway, what is the mechanism of action, at which point should we modulate the pathways that we are interested in, what kind of genes are being activated. And for this, we use omics data, which is metabolomics and transcriptomics. We use bioinformatics to understand a little bit more about what's happening inside the are given to the plants. We also approaches to design or understand what kind of molecules would be useful. So sometimes, if we do not know anything about a problem at hand, we would start from a high resolution approach, get information from bioinformatics as to what kind of molecules would be beneficial, and then look at a high throughput approach to screen these molecules. Now, I would like to take you through the different approaches or different mechanisms that we are using. So, one of the primary approach towards designing biomolecules that we do is use plant signaling. So, plant signal is nothing but a plant's internal communication system. It tells the plant what is happening in its outside environment, and these are like signatures. So a bouquet of biomolecules is produced, and it acts like a signature for a particular type of stress. The plant is able to understand whether it is facing heat stress, whether it is facing a disease or a pest, and these signals are translated inside very rapidly. To be played so that we can see how rapidly these signals are translated from one end of the plant to another. So this is a caterpillar is eating the leaf of the plant, and you see the signal is being started at one end, and it quickly translates inside the entire plant, even when the leaf is detached from the plant. Um, it takes microseconds for these signals to pass inside the plant. Once these signals um, yeah, thank you so much. So once these signals are passed inside the plant, it starts an internal process within the plant wherein chemicals are built up inside the plant, which make it less appealing for the caterpillars, or are toxins for the caterpillars. Bioprime in using plant signaling as an approach is we can have a very targeted biologicals design. They can be very rapid in action, and they can work irrespective of the soil or the climate or other kind of field conditions. So it gives us more consistent result in the field. It gives us more predictable result in the field, and what it lives the farmer with is a more consistent, a more reliable kind of an approach that he's used to when he's using chemicals. Next, yeah. Next, I'd like to talk about the discovery tools. So, I mentioned that we use phenotyping as an approach to rapidly screen the plants. So, a multispectral analysis is used. A multispectral analysis is nothing but seeing in see. So we see only in the visible spectrum of the light. If we were able to shift and look at the plants in infrared spectrum that we currently do not see, we are able to see, visualize using this as cameras very rapidly what kind of stress the plant is seeing. Usually, this happens even before these symptoms are manifested in terms of discoloration, in terms of actual phenotypic variation that we can visibly see. So, multispectral analysis offers us a very rapid tool to see what normally we are not able to see. So, it's seeing more than what meets the eye. To decipher what is the mode of action of these we use transcriptomics. So when transcriptomics is given, when the lead molecule or the microbe is applied, then we isolate different the DNA at different stages, and then it is compared for which genes are amplified at what stage, which genes are amplified in more quantity. So it's a time as well as a quantity analysis that we do. Once we have that done, we are able to understand route that this molecule takes to give the desired result. For example, what is the route that it is taking to give heat tolerance to the plants? In BioNexus, we work on an approach of a microbiome instead of a single microbe. So the approach here is based on a concept called hologenomics, which means that there is more to the plant than just environment interaction. We all know that the genes gives rise to different traits in plant. We also know that these genes are expressed differently in different kinds of environment, and that is currently what the entire breeding or the entire plant variety development approach is based on. But beyond that, the fitness of the plant is determined by what kind of microorganisms it harbors. And therefore, hologenomics is a concept that says that the fitness of the plant is determined by a sum total of entire microorganisms and the genome of the plant in combination with the environment. So in BioNexus, we are trying to look at different kind of microorganisms in the phytospheres, that is, microbes present above, looking at different kind of microbes that are present in the rhizosphere, that is in the soil underground. And here we are looking at not just one microorganism, here we are looking at what is the community of microorganism that enables the plant to withstand different kind of traits. So this is a shift in an approach from a single microbe to a microbe community, or engineering the communities within the soil as well as within the plant. So under BioNexus, our first step to do this would be to build a library of such kind of microorganisms. Currently, we have 6,500 microorganisms looking at. So, looking at the entire approach that I just described and looking at an entire shift in trend, we can see that we are now looking towards system biology, which means that now the biologicals that are coming out are from a more holistic approach. And this is completely opposite of what the synthetic chemicals are developed with. We believe that this holistic approach will give a more sustainable way of managing crops, a more holistic way of managing crops, and leave the soil much better. So, with our SNIPR biologicals, we are able to address climate change and give more productivity to the plants. We are able to very specific time points in a very specific and controlled manner, which gives farmers repeatable results. Currently, we have three classes or three buckets of product with us, which is for stress management, for growth, and for yield. And with the wheat example, I explained to you how they are used to mitigate different kind of stresses. We've reached over 1 lakh farmers and are working in 8 states in the country today. I just wanted to also show you, besides the result on the field, how these products are different in terms of look and feel. So on the extreme left, you can see some current products that are there in the market. They usually black, sticky kind of liquids. They're difficult to mix with waters. And on the right hand side, we have products from our portfolio, which are clear, highly water soluble solutions. These entire products, because they are based on modulating plant processes which are conserved, which means which are same, whether it is a vegetable, whether it is a coconut plant, or whether it is exotic plant. These processes are conserved, so our products are crop agnostic, but they're stage specific. And currently, we work with more than 35 different crops. I want to share just two case studies of how farmers have been impacted by use of these products. So, this is a farmer from a grape growing, and these farmers are extremely progressive. They're very diligent on collecting data. They have sensors in their fields. These sensors send data to their mobile phones. They have weather stations in the field. So they're extremely diligent farmers. But what happened in pandemic is that these grape farmers were not able to harvest the grapes at the correct time, and the grapes stayed on the vines for longer. If you know and are familiar with the grape production, you will immediately understand that what happens is these grape draw in more nutrition, which comes at a cost of the next farmer was already expecting decrease in production. He was expecting a lot of early bud necrosis, that is loss of flowering and fruiting, and he was expecting very little quality produce that he can export. And therefore, these farmers started working with us, and with an entire program that we gave them on biologicals, this farmer was not only able to meet the quality. You can see the difference in quality in terms of the bunch, the size filling, but also in terms of the bricks content that is required for export grapes production. He was also able to earn extra profits. He earned almost 1.5 lakh rupees extra than what he would have normally. Mind you, this was a farmer that was already expecting a loss in yield, but he got more than average of his production. Um, we see that we have very marginal farmers, and this is one such marginal farmer from South India who grows green gram. And we had multiple spells of rainfall, extremely drawn out rainfalls, and a lot of unseasonal rain that led to very little crop growth. And at the later stages, the rainfall impacted the maturity of the pod. The leaves were infected with virus, and there was not enough food that the plant could produce, and the harvest was impacted. Here, you can see on the right side, the beans are mature, they're brown, and they could be harvested. If you take a look at the leaves just below, discoloration. This is how, even in the most severe conditions, targeted physiology modulation can help overcome the most severe of challenges. For this farmer, we were able to reduce his crop loss, and he was able to get almost 50% increase in yield. So across the spectrum, whether it is a progressive farmer or whether it is a marginal farmer, these biologicals can deliver results. Just to give an example for one of our product, which is for growth enhancement, I would like to take you through just a couple of slides to show you the difference in chemistry. So this is a biological for growth. However, it does not contain any hormone. It does not contain any PGR, and yet it is a very defined composition. Its mode of action has been elucidated, and we find that oxygen response pathways, cytokine response pathways are amplified, and they work at a very high efficiency. So even with hormones, we are at a 3x more efficiency. And it upregulates antioxidant pathways, it upregulates photosynthetic pathways. So we now understand exactly how this product is able to perform, and gives us insights into how this product is to be recommended on field. If we look at stability studies, so this on the left hand side, you can see three batches that were produced and over 3 years, and its comparison to a spectral signature that we developed for each of the product. And you see that there is a very high batch to batch adherence. That means no matter how many batches you produce, you will end up with the same chemical composition. And on the right hand side, you will see 4 years stability for a single batch. So a single batch that was produced and assayed over the 4 years, you will see that there's an extreme high stability for that. Now, how does this compare when it is used with different kind of agrochemicals? Because farmers usually make different kind of tank mixes to avoid labor cost. So it's important to see that we do not result in low compatibility. So we've studied compatibility for all fertilizers, micronutrients, and we see an extreme high adherence in terms of compatibility. Finally, what is the impact for the farmer? What is the impact that the farmer sees? The farmer sees a better developed crop, a faster developed and established plant. There's high photosynthesis efficiency, and therefore we see that more flowers are there. In case of plants where there are more female flowers and where there is a male flower and a female flower, we see that in stress, female flower abortion is high. So this product is able to fix those issues, and by increasing per fruit or per grain weight, and thereby contributing to per acre or per hectare productivity. Finally, I would like to say that all of the work that we do is possible because of a very vibrant, diverse team that we have. We are three co-founders who have all done our PhD and postdoctorates in plant physiology. We have a team who has been working on biomolecules, biomolecule identification in different countries all over the world. In addition to this, we have agronomists, agrochemists, biotechnologists, nanobiotechnologists, bioinformatics, molecular biology people who are a part of the team. Over the years, we've been fortunate to win several awards, national and international. Work, especially the development work, was possible due to government funding. Finally, I would like to leave with the thought that we are developing biologicals with the aim of living a better world, not just for us, but for the coming generation. Thank you so much for this opportunity and for allowing us to share the work and the research that we do.
Uh, Thank you very much, Dr. Renuka, for your wonderful and detailed presentation about Bioprime solutions, which were very informative. And I must say that it was an eye opening session to learn the impact of climate change in agriculture and the solutions you're offering from Bioprime by leveraging the biotechnology platforms like SNIPR and BioNexus, which are sustainable, low carbon print, and also the residue free. You also nicely explained the different approaches, particularly like a plant signaling for finding new molecules for discovering next generation ag biologicals, and how these products are very unique from the existing one in the markets. That's really kind of commendable from our end. And you also shown some successful studies, particularly on grape and green gram farmers, and how your products are very helpful in doubling the farmers' income by using your products for progressive as well as the marginal farmers. Your results are really kind of encouraging and positive to the farmers, and I hope it will have a bigger impact on increasing the productivity across all the crops and regions to solve the food demand and make agriculture more sustainable one, particularly for small scale farmers, which are the main beneficiaries, particularly in APO member countries. Thanks again for sharing your thoughts with us. Now we open the floor for questions. And yes, let me start from my end. And actually, I need to go a little bit more detail about the necessary or necessity of going for technologies to address the climate change. Why do we need that? Maybe it is a very basic, but I would appreciate your comments on that.
Yes, thank you so much, Asai. It's a very good question. So, what happens is with climate change, the farmers are not facing just a single stress. Multiple different stresses occur in the single crop cycle. So, Just to give you an example of apples, this season itself in India, at the starting, the apple growers started with extreme high, snowfall. Then there was a water shortage, and now as the apples are getting closer towards size and color development, they are facing heat challenges. So here we look at and we understand that in a particular cycle we probably have to go and mitigate every type of stress that is known or studied academically. Our current agri inputs, which are fertilizers, micronutrients, they work only if the soil is devoid of these nutrients. Hormones would work, PGRs would work if we could, if we only wanted to accelerate certain processes. But what do we do if the processes themselves are not working to a great extent? When we studied, we found that sometimes the plant processes are working at only 40% efficiency. And at that efficiency, even if you add more of our current agri inputs, you're not going to see a result. And that's what's happening, especially in Southeast Asian countries. We see the farmers going back and adding multiple rounds of fertilizers, multiple rounds of micronutrients, though they are not at all required if we do soil analysis and understand. And therefore, new technology is required to address these problems.
Thank you so much. I'm pretty much agree with your point that definitely technology is necessary, especially when we face climate change related, like what you shown the Apple as a case study. Thank you so much. And my next question is from one of your slides, you have shown the trends in biological research, especially ag biological trends. And you've shown that India is a kind of a fourth ranking. That then, but I don't see any other APO member country other than India, particularly Southeast Asian countries. What is the trend actually? How this kind of technologies can be adopted, where we have a more small marginal farmers. How this trend or how the industry is going beyond this kind of ag biologicals? Maybe you could share more insights on this, please. Yeah.
Definitely, definitely. So, just to begin with, the data that I shared was analyzed by using uh, PubMed as a source, and what we did is we analyzed the number of papers that come out of when we give a specific keyword. So, for example, papers that address biotic stress, papers that address nitrogen use efficiency, papers that address microbes, and then kind of did an analysis on what countries they're coming from. So this is a representation of the research work that is going on in different countries, or to say the published work that's there in the domain. But when we look at the new trends that we see, earlier, at least 5 or 10 years before, we saw a lot of research happening on seaweed extracts. We saw a lot of papers that had humic or fulvic acids as their components, active components. But now we see more finer characterized molecules, or we see more different approaches. So we feel that in the next 5 to 10 years, though in the market we see a lot of dominance of humic acid and seaweed based products, we will see more differentiated, more science backed products coming into the market. They could be something like what we work on, which is signaling molecule based or physiology modulation based approaches, but they could also be specified protein extracts, specified secondary metabolites, or they could be different kind of microbes. So we see an opportunity of science-backed products coming into the market and dominating the market in the next 5 years.
Thank you so much. I truly understand that going forward, when we have the kind of science-based technology, then definitely you will have the unique opportunity to go into the market to offer the sustainable solutions. That point I am very pretty much agreed on you. Thanks for that insights. And now again, my next question is. What kind of new tools or techniques will contribute towards the next generation of ag biologicals? And also, maybe you can give more detail about what does exactly the ag biologicals covers.
Sure, sure. So, Ag Biologicals is a very broad umbrella and would cover any kind of bio based product, whether it addresses plant health, plant nutrition, or plant protection. It could also be broadened to include bio based weedicides and herbicides. So, it's a four domain encompassing umbrella for biologicals. What techniques we think would be more used for discovery would be more of automated kind of an approaches. So people want to develop techniques so that they can automate the discovery process. If you see, the discovery process itself takes 5 to 6 years, and then the product validation takes another 3 years or something. So we look at a very long product cycle before any product is commercialized or can be commercialized. Besides this, the cost of development is also very high. So, if you are able to develop high throughput approaches, if you are able to bring down the cost of discovery, those kind of techniques would be something that would be very valuable. So, if there are any startups out there, who are wanting to work on development of these techniques, they should consider developing platform technologies that enable not just their product development, but also can be valuable for other companies to partner with them and develop new kind of biologicals. Besides this, we see a lot of molecular techniques coming into the play. So we see a lot of micro engineering for specific product formation. So synthetic engineering or synthetic enzymes being produced. Those kind of techniques we see, coming a lot into agriculture. However, I think there are two things that we have to keep in mind. No matter what high-end technologies we are using for development, we have to be very aware of the price that the farmer can pay for these products that we do. So anything that we do cannot be extremely pricey, making the products unaffordable for the farmer. So scale has to be taken into account.
Thank you so much. Yes, I agree with you that whatever the new technologies are coming or going to, or the companies who are working on the new discovery molecules, it should be affordable for our farmers, because most of our farmers are like a small scale farmers. If we can't cater that segment, if you take the base of pyramid, most of our 75 farmers, 75% of a segment of coming from the marginal scale farmers. So when we can't cater them, definitely the technology cannot be a successful one. I truly agree on that. And likewise, what you have said, this should be price affordable for farmers. At the same time, also it should be kind of unique as well. So I'm just trying to connect the technology or the solution products which you are offering, how unique it is from the existing ones. Could you please give more insights on that from that perspective? Yeah.
Sure, I'd be very happy to. Yes. So, like I said, our entire philosophy of developing the product itself is very different. We look at it from a more holistic approach rather than addressing just the symptom. The chemical alternatives out there usually are very symptomatic in treatment. And we are trying to look at the root cause. What is it that is leading to the symptom that we see in the plant today? And thereby, the products that we develop become a little more holistic. Our approach of using plant signaling molecules is also very unique. There are very few companies globally who've been able to isolate these molecules, stabilize these molecules for an extended period, and manufacture these molecules on a very large scale. So I think that gives us a very good edge in terms of the SNIPR biologicals. When we talk about the microbes, as I mentioned earlier, we look at the microbes. We have NPK kind of microbes, which is nitrogen fixing and potassium and phosphate solubilizing microbes, and then we have mycorrhiza and trichoderma, and that pretty much sums up the kind of microbes that are very extensively sold in the market today. What we're looking at from a BioNexus perspective is engineering an entire community. It's in terms of looking at the microbiome and trying to see if what kind of microbes the plant requires for its better functioning. So, what are these microbes that associate with plant and are able to confer some traits to the plant? Our approach towards developing microbe-based biologicals is also very different. So, in both these segments, we are able to get very differentiated outputs. I also showed in terms of our sniper products how they look and feel also very different from the existing products.
Thank you so much, Dr. Renuka. I think that was a pretty much straightforward for my questions. Now, as a startup, I would like to understand from your perspective, when you are trying to address the very important global issue of climate change. Now, every nook and corner, you can see that where even recently the concluded on the COP 27. And so here we keep on talking about climate change and how what kind of solutions we need to offer, or how we should address or face something like that. From your perspective, when you try to like address or giving the product from a climate change perspective, what kind of support you are expecting from your own government, or at a government level, what kind of assistance support you need as a kind of startup when you try to address a big global issue?
Yeah, that's a very, very important question that you asked, Asai. Unfortunately, bigger problems like this, which we're trying to solve using deep tech, which means that we cannot start out of a garage, right? I mean, there is a lot of infrastructure that is required for us to just begin and test the products. And the risk of failure is so high that who puts up the infrastructure for the initial testing purpose becomes one of the major roadblocks for more and more researchers like me to transition from academics into a startup sector. Thankfully, our government has been working on developing biotech incubators. So currently in India, we have, I think, 10 or 15 biotech incubators, which are like plug and play kind of centers where people can go, use the modern facilities, and just pay rent basis for the instruments, high-end instruments that are required. So one is, of course, facility and infrastructure kind of a support. Our country also thankfully runs a ignition program where a seed grant is given just to test out the product or the hypothesis. So a proof of concept funding is given. So it's a very, very welcome step, and I'm sure we would not be here had it not been for this kind of an approach. As we move ahead into the journey of the product development, more support is required in terms of testing it out on the field. and that becomes one of the major roadblocks even right now that we face. Access to farmers, access to agroclimatic zones, access to different kind of areas where these products can be tried, tested out rigorously is something that is required. And in terms of policy, a policy change is also required. Currently, government pays heavily for the subsidies that it is given to the fertilizers. So can it not, look at giving such kind of products where the fertilizers use can be brought down by 20-25%? So some kind of policy level rethink is required.
Thank you so much for like expressing your views on the kind of support you need from your government. On the same point, I would like to understand when a kind of startup, when you want to go beyond India, for example, to reach other countries where your technologies really demand. So how you want to like go, and what kind of approach you need, or as a like an international organization like APO, what kind of support that you may require, or so maybe we would like to happy to hear your comments on that.
Yes, that's a very important, I think, bottleneck that you've highlighted here. So the products may be globally relevant. So how do we get out of India and go towards other countries? So our approach here is two-way. What we're trying to do. One is trying to expand our reach into countries which have similar regulatory environment like India's, which accept regulatory data that we've already generated for our registration purposes, and also look at countries which have similar kind of climatic conditions like India, where we already know that the product works and can be introduced into the market. So this is a shorter entry model that I would say. On the other hand, we look at markets which are more mature, like the APAC market, the European market, or even the South or African market, or a South American market, which are very very promising. But here the regulatory requirements are different. And what we like to do is tie up with organizations that can give us an entry, give us a sense of what kind of regulatory is required, help us do the required documentation process, and give us some kind of market access. So there are certain channel partners that we work with, who are our go-to-market partners, who already have an established distribution network outside India, and by an extension of the product working here, help us get into different countries, and that would be a association-based model, I would say.
Thank you so much for your explanations. I truly understand that you need more kind of a regulatory kind of a support from international organization like us, so that you can reach other countries where you have a similar agroclimatic conditions, or maybe the similar kind of practices, so that you can easily enter. We wish you all the best for your expansion of business outside of India as well. I think we are already running out of time. We have reached time, and just as a last question or comment, what could be kind of your key takeaway message for our audience by joining this APO talk?
Oh, yeah, that's a very good pointer that you have raised. So, what I'd like to, say that is a climate change is going nowhere. We have to adapt to it and make our food more secure. There are better ways of making our food secure, and which come not at the cost of the soil. So there are technologies, there are possibilities of reducing carbon footprint, making food nutritious, ensuring that the soil is healthy, and still meeting the food security demands. We as consumers need to show preference for it, because ultimately it is our demand of having more residue free product, more nutritious product, and product that is grown more ethically, that would drive companies to adopt more healthy practices of making or cultivating the foods. So I think we as consumers should emphasize our right, and help driving adoption of such kind of technologies. So be aware that these technologies exist, be aware that these technologies work, and you as a consumer, please demand for food that is grown in a more ethical way.
Thank you so much. Well noted on your last message. And yes, thanks a lot for joining today for this very important productivity talk series. And thank you so much, Dr. Renuka, again for your valuable insights by showing the Bioprime solutions, which are we do understand that this is very sustainable and also targeting small scale farmers. Dear viewers, thanks a lot for joining today's session. And this APO productivity talk is will be held every week, featuring leading experts on different domains. So please subscribe this APO channel and stay tuned. Thanks a lot. Bye bye.
Thank you. Bye bye.